हेलो गाइस, कैसे आप सब? मैं करना भी सिखाते हैं रोटी। इसका हम लोग का पॉइंट ऑफ रिव्यू है मोस्ट इंटरेस्टिंग क्वेश्चन ऑफ एलगी ऑफ chalmydomonas से, लेकिन आप कैमरा पर रिप्रेजेंट कर सकते हैं। So let's get started without any doubt. Systematic position of chalmydomonas, cell division, chlorophyta, class Chlorophytaceae, order Volvoc, family Chlamydomonaceae, genus Chlamydomonas, algae, occurrence. Chlamydomonas is one of the simplest unicellular freshwater algae. It is about 325 species. Occurs in freshwater ponds, lake tanks, which is rocks in nitrogen matter. It provides green turgidity of the ponds lake. There are some terrestrial species which are found along the branches of lake, ponds, and mud soil. It is also found in strangest water of ponds, lake, and ditches, water tanks, sewage tank, and running water. Chlamydomonas cell is a planktonic algae and makes surface of water appear green. Chlamydomonas cell is also found as cryophyte that is growing in snow. Example: C. nivalis causes red due to presence of pigment. Hemiochromo and C. yellow stones is impart green color to the snow. Thallus structure of Chlamydomonas cell is a unicellular motile algae. The cells are biflagellate, spherical, elliptical, pear-shaped. The anterior end of the cell is usually papillate, and posterior end broader, that is C. chordata. The posterior end is pointed. Glycoprotein-rich hydroxyporine are the main structure present compact cell wall. Most of the species of Chlamydomonas are massive cup-shaped parental chloroplast shape, that is a shape C. Biciliaria, described in C. alpina, reptical in C. rectula, ridge in C. steni, axial in C. irradians. C stand for Chlamydomonas cell. The chlorophyll has a single pyrenoids, but in C. calaminae, some species there are more than two pyrenoids are each chloroplast. C. rectula is a chloroplast or not out of the pyrenoids is a protein body in a chloroplast of algae that is involve a carbon fixation and a starch formation. The cup-shaped structure are formed by chloroplast anterior part of the filled with cytoplasm. At the pointed ends of there are two contractile vacuoles which are contracted and osmoregulation of the cell. Each has an eye spot or stigma which lies near the anterior ends. The anterior papillae part of the cell bears two flagella which are equal in length. The flagella are of which plus-shaped type. The flagella are associated with neurometer apparatus of the cell. The neurometer apparatus consists of three main parts: blepharoplast, a small granular structure present on the base of each flagella; paradomus, a transverse filament of connecting the two blepharoplast; rhizoplast, a thin and soft descending thread connecting on the blepharoplast with the centrosome of the nucleus. Reproduction of Chlamydomonas cell by zoospore. Zoospore are found under the favorable condition at the sometime zoospore formation of the wall without any flagella and because non-motile. The contractile nucleus disappear and protoplasts withdraw from the cell wall and divided into longitudinally into two daughter cell. डिवाइड करके longitudinally डिवाइड करके दो daughter cell बनाता है. This is formed by successive longitudinal division. ये successive longitudinal division फॉर्म करता है जो कि राइट एंगल एक दूसरे करता है, फॉर्मिंग 4, 8, 16 और मोर यूनिन्यूक्लियेट प्रोटोप्लास्ट. 4, 8 और 16 फॉर्मिंग करता है और उससे भी ज़्यादा प्रोटोप्लास्ट का फॉर्मेशन करता है। विदाउट द पेरेंट सेल वाला बिना किसी पेरेंट सेल वाले के जियोस्पोर आर लाइब्रेटेड फ्रॉम द प्रेजेंट सेल एंड जियोस्पोरेंजियम जिलेटिनियस और रप्चर सेल वाल जियोस्पोर लाइब्रेट करता है विदाउट प्रेजेंट रहता है जियोस्पोरेंजियम में बाय जिलेटिनियस वाल जो कि रप्चर होता है। अंडर फेवरेबल कंडीशन द फॉर्मेशन ऑफ जियोस्पोर कैन टेक्स प्लेस बाय 25 आवर एंड फर्स्ट वन इज डिवाइड टू फॉर्म टू स्पोर जियोस्पोर फॉर्म 8 और 14 cell wall जियोस्पोर फॉर्म cell wall by planospore. These species formed during unfavorable condition. Under condition of duff, the cell withdraw flagella and their protoplast divided into 2 to 6 daughter cell protoplast. Each protoplast cell secrete a thin wall but doesn't develop flagella. The non-motile cell wall is called a planospore, which also seen in form of image. This is called a planospore under unfavorable condition to redivide to form 8 cell 7 cell after redivide to form a planospore. After redivide to form in favorable condition Moses to form a planospore by hyphenospore. In exotone and favorable condition protoplast develop thick wall and structure development is called haplonospore. Example C. nivalis. The haplonospore also germinate like a planospore on approach of favorable condition. ये सेम टू सेम फेवरेबल कंडीशन में जर्म करता है जैसे कि हाइपनोस्पोर किया था, जैसे कि हाइपनोस्पोर जर्मिनेट करता है जैसे कि प्लेनोस्पोर किया था. द पालामिला स्टेज अंडर फेवरेबल कंडीशन प्रोटोप्लास्ट ऑफ द प्रेजेंट divide to form 4 to 8 daughter cell. The present cell wall of gelatinous to make a mucilaginous sheath and round daughter protoplast. The daughter protoplast also develop a gelatinous wall around themselves but doesn't develop flagella. These protoplast segments are called as a palmillospore. When a favorable condition return the gelatinous wall divide to palmillospore, develop flagella and a spore of the related and to make a new thalli geospore. Palmillospore divide to form geospore occurs to five condition favorable conditions cell wall palmilla stage. Sexual reproduction isogamy type. The sexual reproduction start with the division of the cell wall protoplast 16 and 32 or 64 by flagellar gamete. The gamete are naked without any cell wall. In many isogamous species, a vegetative cell directly functions as a gamete without any undergoing and destruction. Isogamy species characterized by structural similarity between two fungi gamete. The homothallic species of gamete are produced in the same plant cell fused to form zygote. Some species in heterothallic species of gametophyte cell wall two different fuses to form morphology and some species. The flagella of gamete are covered by agglutination of chemical substance involved the recognition of gamete of the opposite strain. The substance are not present in the flagella of the vegetative cell. In heterothallic species, when the gamete plus strain comes in contact with those minus strain, the of two opposite strains adherence because the agentis initially the gamete clamp in a group of 50 with flagella toward the center. In each clump a number of plus or minus gamete in variable. Eventually gamete of opposite strain fuses in pair of sub at the anterior end. The flagella become fuses the paired gamete strain away from the clump. Morphologically similarly because it is called isogamy. And isogamy means in reproduction of gamete are under equal in size. and morphologically same and physiologically don't same and isogamy. The male or female gamete are smaller, and the female gamete or microcyte are larger. Some species of example: microgamete form the male gametophyte, in which protoplast is divided from 8 to 16 gamete wide only. Microgamete are found in female gametarian, which are protoplast divided from 2 to 6 gametophyte. The microgamete are more than one microgamete. The microgamete comes closer due to microgamete. The protoplast of microgamete enters into macrogamete, and after fused to form diploid zygote is formed. The zygote secrete a thick valve transformer into zygospore. Similar size, fused to form 2 to 4 6 male and female to form after divide to form 8 to 16 cell. Oogamy, they are morphologically and structurally same. The oogamy sexual reproduction takes place by some species. The vegetative thallus function as male and female cell with flagella, and directly function as non-motile microgamete or example. The male gamete contain many pyrenoids. The microgametes are formed by 4 division of protoplast in case of angiospermic reproduction, and isogamous reproduction. The microgamete reaches to form female gamete and united by anterior ends. The contact wall between two dissolve after plasmogamy and karyogamy. Diploid zygote is formed. The zygote secrete a thick wall of transformation, zygospore, oogamy, non-motile and motile. After redivide to form calyptogamy and calyptogamy, meiosis division, first division after to form 4 cell, after meiosis second division to form 16 cell, meiosis 32 after gamete. So next is coming, and this topic is end. Share with your friends.